Bone is a highly dynamic living tissue that will undergo a continuous cycle of modelling and remodelling throughout the human lifespan in response to the daily mechanical loading that it is subject to. Bone-resorbing cells known as osteoclasts dissolve damaged bone tissue, paving the way for osteoblasts, cells that form bone tissue, by secreting matrix proteins and transporting minerals to the new matrix. As children, our rate of bone formation greatly exceeds that of resorption, resulting in an enhanced bone healing capacity. However, as we age, our osteoblasts become less active, resulting in a loss of bone density, which makes them more prone to fracture, a condition known as osteoporosis. But what if there existed a way to reprogram adult cells to behave more like child cells for the purpose of improving their bone healing capacity? One possible solution is to incorporate tissue engineering strategies that rely on the interplay between progenitor cells or stem cells, a suitable environment such as a scaffold to support the cells, and the right biological signaling molecules, a mechanical stimulus to encourage cell growth. My name is Gunbay, and I'm a postgraduate student on the Bioengineering Master's Degree Programme, currently working on the development of a therapeutic platform that will use tissue engineering strategies to enhance the capacity of aged bones to heal critical-sized fractures. In order to develop this technology, I've been working closely with the Royal College of Surgeons' own tissue engineering research group led by Professor Fergal O'Brien, for the development of cell and advanced biomaterial-based strategies for tissue repair and regeneration. Earlier in the year, I sat down with Professor O'Brien to talk about some of the technologies that his group are developing.
Areas where we've been working very closely with the AMBER, Advanced Materials and Bioengineering Research Centre, for example, whereby we're developing nanomedicines that are incorporated into matrices like this scaffold. So essentially now what you've got is the scaffold matrix, which is just like the original scaffold matrix, but it now carries nanoparticles, tiny little particles, which are embedded into the scaffold system. And on those nanoparticles is plasmid DNA, which is encoding for the specific genetic target, for the specific gene of interest.
By comparing the genetic expression of adult and children stem cells, Fergal and his team have recently identified a specific gene that is more active in children, explaining why they have a superior bone healing capacity. In light of this discovery, the aim of my project is to develop a suitable biomaterial that could therapeutically target this underactive gene in adults. Having identified a suitable therapeutic, I was able to use in vitro cell culturing techniques to isolate and expose adult bone-forming cells to the drug. Once I had observed an increase in osteogenic capacity due to uptake of the drug by the cells, I was able to move my project onto the next step, developing the biomaterial.
What we've seen in our lab, that for bone repair specifically, the best type of nanoparticles for bone repair are actually made from hydroxyapatite. So hydroxyapatite nanoparticles, hydroxyapatite, which you find in native bones, can be used to deliver genetic cargoes, and that's where our primary focus has been and will be over the coming years.
Using optimized recipes and in-house freeze-drying techniques, I was able to create a scaffold composed of nanohydroxyapatite and type 1 collagen, which is a major component of organic bone. The goal with this was to mimic as closely as possible the native cellular environment within the bone tissue, while also providing an adequate means to deliver the drug. The potential of this platform to promote osteogenesis in adult stem cell populations is still ongoing, but so far the results look promising. So why exactly is this research significant? Currently, the gold standard treatment for large bone defects is surgical autografting, which transplants healthy tissue from one area of the patient's body to the site of injury. However, it is now estimated that 12% of the Irish population are over the age of 65, and with this comes an increase in the incidence of bone injury and subsequent lack of suitable donor tissue. What my research team and I hope to achieve with this technology is a viable alternative to conventional bone grafting, whereby therapeutically loaded and easily reproducible biomaterials such as this one could be implanted at the site of injury in patients who have a reduced bone healing capacity. Once implanted, the scaffold recruits the patient's own cells to the site of injury. At this point, the cells are exposed to the therapeutic molecules, significantly increasing their natural osteogenic and bone-forming capacity. After some time, the bone-mimicking scaffold degrades as the newly formed bone takes its place. Thank you for watching my video.